I'd like to invite the lead author, Professor Norlisah Ramli, to present their paper.
Thank you, Prof. Go. Assalamualaikum, Bismillahirrahmanirrahim, and good morning. I'll be presenting representing the author for the Faculty of Medicine Best Neuroclinical Paper 2018. The title is Quantification and Visualization of Lipid Landscape in Glioma using N-nanopose phase. The authors are Dr. Po Chu Xiao, Dr. Jenny Wong Siu Ding, Dr. Aditya Hernowo, Professor Vairaban, and myself, Professor Norlisah Ramli. This is the introduction. Glioma comprises 27% of all brain tumor. Glioblastoma is one of the most aggressive form, and the prognosis of GBM has not significantly changed over several decades. The median survival remained at 12 to 15 months. This is despite advances in radiosurgery, radiotherapy, and chemotherapy. Therefore, genomics, proteomics, and lipidomics have been employed to discover potential biomarkers and therapeutic targets. In fact, WHO guideline upgrade in 2016 recognizes this fact and integrated molecular parameters into the grading of gliomas. A little bit of background of lipids and glioma. K. Gopal have found that the glioma have higher lipid content than other tumors. Poptani have observed that lipid resonance in MR visible and invisible necrosis in majority of high-grade gliomas and not even in a single case of low-grade gliomas. P.S. Murphy shown that lipid levels does correlate with high-grade and low-grade tumor status. We have done a systematic review looking at quantitative MR imaging and radiogenomic biomarkers for glioma characterization. And the summaries on your right side, so it shows that Guo et al. Found that increase in lipid biosynthesis has been recognized as one of the essential elements of metabolic reprogramming in cancer cells. Outwater found that IOP, this is what we are trying to investigate, has been used clinically for lipid detection in disorders such as those listed here: adrenal adenoma, angiomyolipoma, fatty liver, and bone marrow pathology. Ramli et al. Found that elevated lipid level has been found to correspond with higher tumor grades and aggressiveness of brain tumor. However, few studies located at lipid quantification for gliomas. Why did we choose lipid? We found that exaggerated lipidogenosis is an essential element of metabolic reprogramming in cancer cells. It supports rapid proliferation of cancer cells to fulfill the increase in demand for energy and nutrients. There's a lot of lipid in the brain. Those are listed here. Lipids have an important role in apoptosis, necrosis, signal transduction, and attributed to proliferation, inflammation, malignancy, growth arrest, and migration and invasion. So what are the problems that we face currently? The glioma is very complex and heterogeneous. There's no visualization of lipid landscape in glioma. So far, for non-invasive lipid quantification method, there's only MR spectroscopy. There is a need for an easily available non-invasive imaging biomarkers that reflects lipid distribution to improve the clinical assessment of glioma. Therefore, there's limited imaging prognostic markers for evaluating survival outcome. So what we are trying to do is moving from structural MRI, which are conventional, into more physiological MRI to tell us a little bit more about lipidomics that's happening in glioma tissue. The research objectives are to evaluate the feasibility of IOP, the in-a-post phase, in lipid quantification of glioma, to quantify the biomarkers lipid fraction in discriminating the rates of glioma, to visualize the lipid distribution within glioma for diagnosis and grading, to investigate MR lipid quantification for prognostication. These are the equipment that we use. We use the 3T MRI scanner, which is in the hospital, to perform the structural as well as the physiological MR. The material we use are the lipid, for the lipid phantom study, are the lipid infusion used for intravenous parenteral nutrition. These are the software we use, which is the Osiris, ITK-SNAP, MATLAB, SPM, and the LC model for MR spectroscopy. This is the gold standard that we've used for fat suppression that is available in MRI. Most of the time, we use chemical shift-based water-fat separation. However, it takes a very long scan time and a more complex reconstruction. It does allow quantification of lipids, as others just allow visualization. We propose an inner-post imaging as another alternative for lipid quantification. The mechanism is a difference in resident frequency between proton nuclei of water and fat in the tissue. IOP provides a whole brain coverage compared to MR spectroscopy and in a very short scan time of 1 to 2 minutes. So what we do, we visualize is during the in phase, within the voxel, you see the water and fat signal together. And when you perform the out phase, the post phase, you only have the water signal minus the fat. So how we quantify the percentage of fat is the signal loss ratio. We take the signal intensity in the in phase minus the signal intensity in the out phase over the signal intensity. Just a comparison of MR spectroscopy and in a post phase, basically to see that the acquisition time for MR spectroscopy for single voxel, just one voxel, is very long, 6 to 7 minutes. While the post phase, we can have the whole brain coverage here, and it's very fast at less than 1 minute. MR spectroscopy, it was the gold standard. It allow you accurate quantification of the metabolites and give high signal to noise ratio. In a post phase, allow shorter acquisition time. It gives you a whole brain coverage compared to MRS and not susceptible to artifact formed by adjacent tissue. Here is our first paper, novel application of chemical shift grade in and post phase in 3T. So this is the first paper that allow us to build on what the second paper which win the FOM Best Award. And here the paper is looking at quantification, lipid quantification for characterization of gliomas using in and post phase. This is a phantom study, and we want to see whether the IOP reflects a lipid quantification. There is two arms to the study. First, we look at the phantom study, and we do some preliminary clinical study using 9 grade 2, 3 grade 3, and 9 grade 4 gliomas. Phantom preparation, we use the intravenous intraparenteral lipid concentration of 0 to 20%. So we dilute it, put it over a little box here into the bottle, and then we perform in, a post, and a post phase here and measure the ROI of the signal intensity. We also perform MR spectroscopy on each of these and do LC model to actually see the quantification. So we have 2 peaks at 1.3 parts per million and 0.9 parts per million. For the clinical study, we perform ROI over the tumor and placement, and then we do the spectral analysis and statistical analysis. So here's the results to see whether there's any linear correlation between the lipid concentration, non-lipid concentration that we have against the MR spectroscopy and also the lipid fraction on the in and post phase. And we found that at lipid 0.9, this is the gold standard using LC model and MR spectroscopy, we got good linear correlation. And also when we use lipid fraction using in and post phase, we have really good linear correlation between the two. So the linear correlation is between 0.79 to 0.99. So IOP does reflect lipid concentration. We then do the clinical study to see the lipid fraction at the solid region was the best measure for differentiating glioma grade using optimal cutoff point VUS. And so we have very good probabilistic classification. For grade 2 is about 100%. You can see they're very far away from each other. Grade 3 is about 50%, and grade 4 is about 89%. So from the phantom study, we found that the lipid fraction were correlated to the lipid peaks on MR spectroscopy and demonstrated that small lipid fraction was detectable by IOP imaging. The relative high signal loss implicated by high lipid fraction is useful to reflect increased microscopic lipid distribution. The clinical study showed that lipid fraction of the solid tumor region was the best measure for differentiating of different grades. The limitation is the lipid fraction quantification was subjected to inter-investigator variability and manual ROI placement. This leads to our second paper, which won the FOM. So here we want to do the whole tumor mapping, and we want to answer the question, can lipid fraction discriminate the glioma grades? Again, can the lipid distribution of the tumor region be visualized? So as I explained earlier, that the glioma is very heterogeneous, and one of the problems that we faced with the previous study is the manual placement of ROI. So we try to get above that by doing an automated tumor delineation using ITK snap, and we differentiate the tumor region mass into solid enhancing, solid non-enhancing part, and also the cystic part. And from here, we get the mass and we measure, put it over the lipid mapping and get the lipid fraction, and then use a lipid map to composition of lipid fraction. And we use image post-processing of lipid map and tumor region mass. Then we perform our statistical analysis using Kruskal-Wallis and Spearman. This is our patient demographic study. There's patient demographic shown just now. There were more patients in the grades, and here we have the identification of tumor region using different MRI sequence of GBM patient. So we use a T1 and T1 contrast to see the enhancing part, the T2 to see the cystic part, and the diffusion weighted to see the solid non-enhancing part, which is over here. So we do the mapping of that, and then we also do the lipid color mapping. The blue depict low lipid fraction, therefore less lipid, and red depict the high lipid fraction, which means there is more lipid. So the results that we found is we look at the lipid fraction in the solid enhancing, and cystic region, and we found that the greater differentiation between the three grades of tumors, the grade 2, grade 3, and grade 4 in the solid non-enhancing region. And when we do a post-hoc test, we found that the greater differentiation lies, of course, between the low grade and the highest grade with high statistical significance. So here's showing the box plot, the distribution of the lipid fraction of the solid non-enhancing across WHO grades, and you can see that we can differentiate between the three grades, 2, 3, and 4 here. Here is showing the color map of the lipid. So if you look at the grade 2 here, the color map of the lipid distribution is quite homogeneous, and they're mainly blue, so they're low lipid fraction. While if you look at the grade 4 here, they're more heterogeneous, they're more blue and white and yellow here. So, and the higher the grade, the more heterogeneous the appearance of lipid fraction, and also the higher is the lipid fraction. So lipid fraction of the solid non-enhancing region is useful for discrimination of the tumor grade because it harbors extracellular lipids from broken cellular membrane. The structural lipid alteration could be attributed to topographical distribution of the lipid molecules due to tumorigenesis. High expression of the lipid is contributed by the prompt release of storage for cell growth and biological membrane generation. Breakdown of the cellular membrane structure in response to growth stimuli resulted from malignant cell transformation, increased number of cytoplasmic vesicles. The results were improved from the previous study. Just to highlight what has happened since then, we're now looking at lipid quantification in predicting survival outcomes of glioma. If you must know, the histology is the gold standard. However, the gold standard is also based on something else, which is the survival. So the grade 2, the histo shows longer survival, while grade 4 is the shorter survival. So we want to see whether lipid quantification can bypass that and look at the survival outcome of the glioma itself. So here we did a similar study. There are more in the 17 patients in grade 2, 8 in grade 3, and 21 in grade 4. We now measure their overall survival and time to progression. And when we do the overall lipid fraction of all the tumor without classification them due to histology, if you look at the volume under the curve here, you can actually see there are ways to differentiate them into 3 categories. So low lipid fraction, medium lipid fraction, and high lipid fraction. And we can obtain the upper cutoff point of 0.07 and lower cutoff point of 0.05. And these are the results of the patient, the demographic. If you look at across, as you expected, the KPS score has bearing on the overall survival and time to progression as expected. So if you have a higher KPS score, the overall survival is longer. And also the WHO grade, the lower the grade, the higher the overall survival and the longer time to progression compared to grade 4. So now we look at the overall survival. We compare WHO grades. Of course, if you compare to grade 2, there are hazard ratio of 8 times compared to grade 2. Now, and also a time to progression is when you compare to histo grade 4 is 6 times faster than grade 2. And when we look at using our lipid fraction, we also find that high lipid fraction has 5 times the hazard ratio to the overall survival compared to the low grade, and 5 times the higher time to progression compared to the low grade. And when we look at the survival curve here, you can see there's some overlapping when we use the histograde between 3 and 2 as what usually happens. But when we use lipid fraction of the solid non-enhancing region, we see better differentiation between the low lipid fraction and the medium lipid fraction in the overall survival curve. Conclusion, the solid non-enhancing region demonstrates significant lipid fraction change in the survival analysis, revising location for biopsy site, which tend to be going towards the enhancing portion. The lipid fraction of the solid non-enhancing discriminate the glioma grades, and lipid distribution can be mapped and visualized to show different grades of tumor, and lipid fraction is a potential prognostic marker in predicting the outcome of patients. What has happened since then? A few more studies has come out looking at lipid role as hallmark of the human glioma metabolism and lipidomic as a promising cancer biomarkers. And sorry, no, there's a few more. Okay. Also highlighting that the non-enhancing contrast tumor is a new frontier in glioblastoma research rather than the enhancing, which was the standard. The future works are imaging and blood biomarker that could reflect lipid profile in tumor, eliminate invasive and laborious laboratory lipid tests. We might want to use computer-aided diagnosis to facilitate diagnosis and segmentation, and link MR imaging and lipidomics for brain tumors, characterization, and untapped areas of research. So therefore, understanding tumor lipid profile, lipid types, and composition towards formulating lipid-based therapy in the future. These are the people that are involved in the overall study, a lot from neurosurgery, biomedical imaging, biomedical science, as well as UMREC staff. Just to highlight that we have an Asian Oceania Congress of Radiology in July 2021. Abstract submission is open now. Thank you, Prof.
Thank you, Prof. Norlisah. That was a very interesting lecture, and I think we're privileged to learn about what sounds like very cutting-edge research in glioma. Questions? Can I have a look at the questions that have been typed in? So far, no questions. Maybe I'll ask a very simple question. It would be intuitive that you think that the enhancing portions of the brain, a brain tumor, would be actually the ones that would show up the differences, right? Rather than the non-enhancing. Any sort of explanation for that?
Yeah, we were expecting that as well, but we found that even though the enhancing part has a higher fraction of lipid, but they cannot differentiate between the grade 2, 3, and 4. So my thing is that maybe the vascular, because the enzyme has a lot of vascular, a lot of moving object, it might not reflect what is truly happening in the metabolomics of lipid at all. So lipid is more like a storage rather than an frontier of what is happening.
What about the cost? You mentioned at the time you save time compared to MRS. Does this incur additional cost of this IOP sort of procedure?
IOP is available on most MRI. It's better on 3T than 1.5 because of you need the signal intensity in that. But yeah, it's available on most because that's been used for lipid quantification in the whole body, in the adrenal as well.
So it's better if one does it on 3T MR compared to a lower Tesla.
Yes, it's only applicable. The chemical shift works better on the higher 3T.
Okay, if there are no further questions, we shall proceed on to the next presenter, who is presenting on the Basic Neuroscience Award in 2018. As you know, the value of the award is actually dependent on the rigorous referee or selection process. So for the FOM Best Neuroscience Paper Award, we actually have external assessors, which consists of senior academics and well-published neuroscience and neurologists from both Malaysia, as well as from Australia and Switzerland for this particular exercise. So the next winner, I will invite Associate Professor Ong Kian Chai, who will be representing the team. And the winning paper was entitled RSAD2. I'm sorry, I cannot give you the the full name. AIM2 modulates Coxsackievirus A16 and Enterovirus A71 replication in neuronal cells in different ways that may be associated with their 5' non-translated region. And this was published in the Journal of Virology. And the first author is Tineshwari Yogaraja, and the lead author is Professor Kamtong Wong. Professor Ong.
Good morning. Thank you very much, Prof. And I represent our team today to present this paper, which entitled RSAD2 and aim to modulate Coxsackievirus A16 and Enterovirus A71 replications in neuronal cells in different ways that may be associated with their 5' untranslated regions. And this team actually consists of Dr. Tineshwari, which is now in NUS, and led by, as mentioned by Prof. Professor Wong Kamtong from the Department of Pathology. Okay, so basically, I will first talk about some introductions about these two viruses. And these two viruses, actually, they belong to the Picornaviridae family, and within the Enterovirus A species, and both is a single-stranded positive RNA virus, which causes hand-foot-mouth disease, HMD. And these two viruses basically show many biological features, such as they have quite a similar structure in terms of their capsids and also their genome organization as well. And just a brief introductions about how these two viruses replicate in cells, because this will lead us to a better understanding later on about our findings. And basically, this virus, as I mentioned before, is a single-stranded RNA virus, which consists of 5' uncoding region, also we call 5' untranslated region, and followed by a capsid protein and non-structural proteins. Basically, the capsid protein will form the outer layer of a virus, and this will allow the virus to bind to a particular receptor that express on the cell surface to allow them to enter the cells and release their viral RNA or their genome, and will lead to further replications. They will first get translated to produce the polyproteins, and these polyproteins later on will be cleaved to produce an individual viral protein, and later on they will assemble. And although on the same time, the viral RNA itself will get multiplied to get more genome in order for them to form a mature virus before they release out from the cells. And the receptor that used by these viruses was SCARP2 as the entry receptor and other reporter receptor as well. But SCARP2 receptor until today is still recognized as the entry receptor for these viruses. Of course, for CA16, Coxsackievirus 16, the actual receptor is yet identified. And about introduction about what is hand-foot-mouth disease. And hand-foot-mouth disease actually is quite common in Malaysia, and it's common pediatric infectious diseases. And usually the patients will have some sores in the mouth and skin and rashes with red spots in their hands, buttocks, and the genital area. And usually the children will present the clinical symptoms will be poor appetite, sore throats, lack of energy, and high fever. And how the virus spread actually is by close contacts, by the blistered fluid example or some fecal material, because the virus can be shed out from the infected children's body. And the preventive measure is quite similar with COVID, I would say, is about close contact and personal hygienes, etc. And although most of the cases, the children will recover within a week time, but however, it's a very highly contagious virus. This is pictures as obtained from Panama. And you can see that in 2018, Malaysia have experienced a quite serious outbreaks of HMD. Just within 7 months, a total of 41,000 cases have been recorded actually. So it's highly contagious diseases. And of course, the symptoms, as I mentioned in the previous slides, is fever and etc. However, there's some complications that are reported in patients or children that are infected by HMD. The most serious one will be CNS complication, central nervous system complication, but this is always associated with enterovirus A71, but not on CA16, as reported in many papers, including our group as well. And our group have also performed some histopathological analysis on the human autopsy samples to further understand the pathogenesis of Enterovirus 71. In contrast, for CA16, as I mentioned before, is very rarely reported or associated with CNS complication. And the first papers that reported the association of Coxsackievirus for fatal infections is actually in 1963. And later on, so the more reasons will be reported in 2009. So most of the time, the Coxsackievirus doesn't really cause central nervous system complications. So we are wondering why CA16 is less capable of causing or less frequency in causing central nervous system complication. These are schematic diagrams that we postulate that how the virus spreads within the body and how they travel to the brain. And as you can see from there, actually the exposure routes or the routes of infection is possibly via tonsil, because we have found a viral infection in tonsil specimen from our human autopsy samples, and maybe from the viruses that shed out from the fecal and from the blister from the skin. And later on, the virus will travel to the blood. That's what we call viremia, means the presence of virus in the blood, and the virus can spread now to all other places in human body, in the organs, different organs. And we believe that the virus entry into the central nervous system is by infecting the skeletal muscles or motor nerve and travel through the nerve to the central nervous system, as shown in this diagram. And is it because of CA16 is not able to travel into the brain, and that's why it's less associated with CNS complication? But however, in our some studies that we have conducted in animal model, we found that actually CA16 actually can be traveled or can be transported into the central nervous system. Similarly, like Enterovirus A71. But however, the infections rate appears to be a bit lower. But the point is, we believe that CA16 is the lower number of CNS complications caused by CA16 is not because incapable of this virus to able to transfer or transported to the central nervous system. And as shown in these two papers, although these two papers just published this year, however, we already have some preliminary data 3 years ago before we performed the studies. And we also have a paper in 2016. This is the foundations of this study. Basically, we compared Enterovirus A71 and Coxsackievirus infections in human neuronal and non-neuronal cell line. What we found that actually EV71 in general replicates higher compared to CA16. That might explain partially why EV71 is able to cause more CNS complications compared to CA16. However, we also found that EV71 actually does not really proliferate so well in neuronal cell line. So that's why we hypothesize that there may be some cell type specific host responses that present in the neuronal cells or in the brains that to restrict both virus infections. And why the response will be different compared to these two viruses. Another possibility could be the genomic difference between these two viruses, although these viruses were quite similar in terms of their nucleotide sequence, about 85% similarity. But this also might contribute to the different host responses that trigger towards these two viruses and leads to disease complications. So in order to study how about a host that what's the host response, so we perform a transcriptome analysis. In this case, it's a very powerful platform that enable us to analyze the full genome or full gene response of human cells in this case. So this platform will be useful for us to investigate what are the genes that are upregulated and downregulated towards a viral infection. And actually, we are comparing two viruses together, but we publish in a different paper. So the first paper we published is actually in 2017. We found that from the transcriptome analysis, we found that AIM2 inflammasome actually is restricting the viral replications in neuronal cell line. I will come back to this AIM2 later on because this paper, the present papers also will investigate what is genomic differences that is resulting in AIM2 expression, but let's focus back to the CS16 first. From our transcriptome analysis, we found that the cell death and antimicrobial responses were upregulated in this case. But however, from our study as well, we found that the survival genes, the cell survival genes were significantly upregulated, not downregulated. What it means is actually the neuron cells, once get infected, they are survived. They are not dying. Okay. So, however, from our virus titration also, we found that the virus does not increase significantly from several time points. What it means that the virus seems not capable to replicate in this neuronal cell line. So, therefore, by using this transcriptome analysis, it actually is a powerful platform, as I mentioned earlier. You can narrow down further what is the genes that are regulated towards the infection by clustering them. In this case, we are more focused to the antimicrobial profile in this case. And we found that all the antimicrobial genes that are regulated basically is from interferons stimulated genes in this case, as listed in this table. As you can see, the fold change is very high compared with EV71. EV71, they are not as high as CS16. So, but although this transcriptome analysis is a very powerful platform, but however, to more precisely quantitate the gene expression, that's why we perform a quantitations real-time PCR, and then we compare the expressions level for these several genes that we have selected. And we found that the RSET2 is the highest upregulated genes in CS16 infected cells compared with EV71 and significantly. And also, RD cells in this case serve as our positive control. And because of it's highly upregulated and it shows very differences compared EV71 and CS16, that's why the subsequent work will only focus on RSET2, not other interferon stimulated genes. So actually, what is RSET2? It's actually an interferon stimulated genes involved in innate immunity and is responsible for antiviral responses and also called virulent. And we choose RSET2 because the level between CS16 and EV71 were the most significant difference. And RSET2 is recently described as an ISG, interferon stimulated genes that have a broad range of viruses, such as dengue, West Nile, and chikungunya virus. That's why we only focus on RSET2 in this case. But however, we cannot rule out the possibility of other genes that are responsible as shown in these slides, right? They have other 6 genes more. So how do we confirm RSET2 is the main genes or the only genes that are responsible for the restrictions that we observe in CS16 infected neuronal cell? Therefore, we perform another method called, we knock down the RSET2 gene by using a system called srRNA. It basically is to destroy the mRNA because mRNA is needed for translate into a protein, and only the protein will function in this case. If we destroy the mRNA for RSET2, theoretically, there's not going to have any proteins get produced in this case. If RSET2 is the ones that restricting CS16 virus infection, after we knock down the RSET2 genes, by right, the virus now can replicate higher, and the RSET2 genes will be lower expressed. So the results show what we hypothesize. As you can see from this row, ACE2 MR level was significantly decreased in the knockdown cells compared with the unknockdown cells. On the same time, CA16 viral RNA copies number will increase significantly. It means that now there's more higher copies number observed compared with the unknockdown cells. If there's a restrictions happens, it will restrict the viral RNA copies number. So also we also quantitate the live virus productions from the infected cells. So what we found that CA16 live virus quantity was significantly increased in the knockdown cells compared with unknockdown cells. So what all these results suggest that ACE2 is a very important gene in this case in restricting CA16 virus infections. And also we compare the cytomorphology of the infected cell. As you can see here, this SKN-SH is the knockdown cells, and this SKN-SH/SI asset is the knockdown cell. You can see they have much lesser cells compared with the unknockdown cell. This highly further confirmed that now the virus seems to able to replicate higher compared with the unknockdown cells. Further again confirm the important role of ACE2 in restricting CA16 virus replications. We now have confirmed that ACE2 is important in restricting the virus, but what's the difference? Why EV71 is not able to induce high expressions of ACE2? That's why in order to, now we go back to the genomic differences. There could be genomic differences between these two viruses. Therefore, we perform some molecular techniques. We produce an infectious DNA clone, in this case, to modulate or to modify the virus so that we could study what is the genes that are responsible for the differences responses that we observe. So basically, we will clone the full genome of these two viruses into a vector, and in this case, we will perform some in vitro RNA synthesis to produce the RNA, and then we transfer back the cells, transfer back the RNA into the cells to produce live virus. In this case, it's a very powerful techniques that we commonly use in biological fields to modify a virus. So to understand the role of the genomic differences, but as you can see, this is the genome of EV71 and CA16. As you can see, there's many different genes that are involved in. Let's say for capsid proteins, we already have 4. So which one to choose? After we performed a literature review, that we found that we think that 5TR maybe is a good candidate to exchange between the two viruses, because 5TR is very important as a translated, is responsible for translations of viral proteins. So, and also reported in porovirus before, is one of the neurovirulence determinants for porovirus vaccines. That's caused the porovirus vaccines to be agglutinated oral vaccine. So we perform an exchange for the 5NTR. So it means that CA16 5NTR now we swap it into EV71, exchange it with EV71 5NTR, and vice versa. And we produce a chimeric viruses in this case. Now the EV71 will contain CA16 5NTR, and CA16 virus will contain EV71 5NTR. And then we perform the same experiments again to investigate whether ACE2 is upregulated or not in the chimeric virus. And basically, the structure didn't change much. Didn't change at all, actually, because we only changed the 5NTR region. So the capsids remain the same for these two viruses. So it won't affect, they're not going to show any differences in terms of binding. So it means it makes the investigations more accurate and precise in this case to determine the role of the genomic difference, the 5NTR differences in this case. As I can see from this graph, firstly, we will investigate the ACE2 the regulations or expressions of ACE2. As you can see, after we have changed the 5NTR, and you can see it in EV71 chimeric virus with CA16 5NTR, the replication, the fold change or the expressions level was similar between the original CA16 virus. Conversely, the CA16 virus now, which contain EV71 5NTR, will similarly show the similar expressions ACE2 level. So this means that the 5NTR is is responsible for modulating the ACE2 expression in this case. So we further quantitate the virus titer, because if it's true that ACE2 is modulating, the 5NTR is modulating the ACE2 expression, we could see a reverse pattern or different change of phenotype from the virus, because originally CA16 should replicate lower now. should replicate lower in the cells, but after we exchange it with the camera with EV71 5NTR, it replicates similarly with the original EV71 virus. Same thing happens when we exchange the CS16 5NTR into EV71. Now the EV71 behaves similarly like the CS16. So again, these results confirm that that 5 untranslated regions was the ones that modulating the RZ2 expressions. As mentioned previously, we also found that M2 is responsible to restrict EV71 replication. Of course, when we first presented, of course, a lot of people does not really make sense because EV71 is considered a neurotropic virus. So by right, EV71 should replicate higher in neuronal cells. But however, from our study, we found that M2 expression actually is responsible for them. So actually, what is M2? M2 is a short form of absent in myeloma 2. Actually it's an inflammasome. What is inflammasome? It's a multi-protein intracellular complex that detects pathogenic microorganisms and activate highly pro-inflammatory cytokines such as interleukin-1 beta and interleukin-18. And inflammasome also will induce a cell death terms pyrotosis. And this inflammasome have been reported if these regulations were associated with a number of autoinflammatory syndromes and autoimmune disease. So this might explain why EV71 partially that cause more CNS complications, although both viruses seem able to be traveled into the brain. So we made use of the construct that we have constructed to study CS16 to investigate how is the M2 expressions now, whether the 5 untranslated region also responsible in modulating the M2 expressions. And with what we found that and the CS16 virus, which supposedly does not upregulate M2 from our previous study, have now become the expression level was similar with the EV71 virus as shown in here. Same happens at 48 hours post-transfections. Conversely, so for EV71s that have exchanged, the camera virus exchanged with CS16 5NTR, the virus now behave like CS16, which doesn't show high expressions of M2 expressions. So this again suggests that and confirm that M2 actually is modulated by the 5NTR regions of the virus. And we're also interested to investigate whether these specific cell restrictions factors is only occur in neuronal cells or it occur throughout all other cell types. That's why we perform using the same constructs to perform in other cell line in RD cells. RD cells actually is a human rhabdomyosarcoma cell line. It's a human muscle tumor cells, and viral cells is a monkey kidney cells, which this cell is very highly susceptible to both of these viruses. So it means that they don't have any restriction factors there. That's why we transfect this chimera virus into these cells, and we did not find any significant difference between the chimera virus and the parental virus. So this further suggests that the cell restrictions factor is cell type specific. It only occur exclusively in neuronal cells, but not in other non-neuronal cell lines. And why there's differences in the 5? What is the role of what caused the 5NTR able to modulate both genes, both restriction genes in this case? So we perform a secondary structure analysis, and we found that actually the 5NTR is, although it's not a coding region, a non-coding region means that it does not translate into a protein, but they can form a certain secondary structure like what we see here. This is the secondary structures for 5NTR for CA16, and the B1 is for EV71. As you can see, there's a missing loop 7 in EV71 compared with CA16. So these differences might explain that might cause the differences in the expressions level of M2 between CA16 and EV71. And as a discussion, so this is a schematic model of how EV71 induced pyrotosis. Firstly, the virus must attach to the cells and enter the cells. So once it's entered the cells, it can sense because cells have an intrinsic sensing mechanisms that can sense the presence of the virus. And Then later on, to aim to get expressions, and it will activate certain genes like caspase-1, and then leads to the release of interferons, interleukin 1 beta and interleukin 18, and release the cellular content and cause massive inflammations. And at the same time, the cells will go through pyroptosis cell death, and the loss of cell membrane integrity will lyse the cells. So this may explain partially is that why in EV71 and encephalomyelitis patients we see very massive inflammations compared with the number of viral antigen positive cells. And these are schematic models of how ACET2 inhibits CS16 infection. And actually, the exact mechanism is still unknown, but we postulate that once the virus enters the cells again, like similarly like EV71, it gets sensed by the host, and then we produce the ACET2 proteins, and the ACET2 proteins will inhibit viral replication. More investigations is needed, actually, to further elucidate the actual mechanisms of how ACET2 inhibiting CS16 replications. As a conclusion from this our study, ACET2 have been confirmed to inhibit CS16 but not EV71 replications in neuronal cell, and conversely, only EV71 induced M2 inflammasome, and it makes its infection in neuronal cells. And this could be due to the differences in the secondary structure of the 5 untranslated region, 5 NTR between these two viruses. And overall, CS16 and EV71 elicits different host responses to infections. This might help to explain the apparently lower incidence of CS16-associated neurovirulence in HMD patients compared with EV71 infection. However, more investigations are needed to further understand the molecular mechanisms of how ACET2 restricting CS16 virus infections. So last but not least, we would like to acknowledge the Ministry of Higher Education for providing us the grants and from the Ministry of Higher Education. And also thank you very much for the faculties on awarding us these prestigious awards. Thank you very much for your attention.
Thank you very much, Professor Ong, for a very comprehensive lecture on your research work. I think there are a couple of questions here. So one of them is, in your opinion, how is the 5' UTR how does the 5' UTR modulate RSAC and other interferon genes? And are there any studies in the role of the 5' UTR in the interferon stimulation and suppression?
Okay, thank you very much for the questions. Actually, from this comparison between EV71 and CS16, actually we don't, more and more evidence show to us that the 5' NTR actually does not really modulate RSAT2 completely, actually, because in EV71, it seems that RSAT2 does not upregulate in this case compared with CS16. So what we believe that maybe because of in EV71 case, EV71 upregulate AIM2 inflammasome. So once it gets activated, so will lead to pyroptosis cell death. So it will cause a lot of host machinery for protein production get shut off, get stopped productions. As a result, the RSAT2 become lower in EV71 during EV71 infection. So we believe that actually the 5' NTR is more responsible for AIM2 expressions, not so much to RSAT2. So that's why, as I mentioned in the slides, further study are needed to fully elucidate the molecular mechanism or the genomic or the genes that are responsible to modulate RSAT2 expression. But from previous study have been shown that it can usually is targeted the non-structural proteins such as 2A, 3C, or 3A. I hope I answered your questions.
Yeah. Any other questions? Can just type. I think we still have time. While waiting, maybe I'll ask Prof. Ong a hypothetical question. How does your research help, for example, in, say, you're making chimeric genes and things like that, in terms of maybe coming up with a vaccine?
Yes. Technically, it is possible to make a chimeric vaccine. I think some our colleagues in the faculty is using this approach as well to produce a multivalent vaccine. But the camera, the challenge will be you need to identify the immunogenic epitopes of the virus so that when it injects or it inject into the animals or into the humans, is able to produce a productive immune response. So on principle, is workable. Is workable and will be high and more cost effective because you just need to have one seed virus to produce the vaccines. Compare you need multiple.
Yeah, I mean, I'm talking about using the Coxsackie A16 to help make a vaccine for the EV71.
Yes, it's feasible, but from some of the studies that the problem now is to identify whether the CS16 vaccine is able to show protection against EV71 because based on the most immunogenic epitopes is usually present in the VP1 regions, viral protein 1, which are at the surface of the virus. And most of the antibody or monoclonal antibody they produce for EV71, sometimes most of them cannot cross-react or bind to CS16. So we believe maybe same way also might happen for CS16 as well, unless you can construct or you can find a conserved epitopes or immunogenic epitope between these two viruses, then this chimera virus will be very useful in vaccine productions or to construct a multivalent vaccine, in this case, against these two viruses.
Well, I think there's no further questions, but you all know who the speakers are, and you can always collar them in the corridor, or if you're worried about physical distancing, then email them. Thank you very much, Prof. Ong, as well as Prof. Norlisah, for presenting two wonderful and very interesting and scientifically rich papers. This highlights the value of the FOM Best Neuroscience publication, and I hope for the next year, which we shall open for nominations shortly, we will get many more nominations. Last but not least, I'd like to thank the technical team from the faculty, Shams and Hayati, for helping us, run this session successfully. Thank you everyone for joining us.